Welcome to my channel Adhyan with Flow. Let's start with enzyme. So we commonly know that enzymes are proteins. Your NCERT said that almost all enzymes are protein. This is the statement, guys. This is the statement that almost all enzymes are protein, but there are huge exception over there, like ribozymes. They prove this statement wrong. RNAs. Okay, guys, listen to me. What are enzymes? The first important thing, I'm giving you most important properties. They have high catalytic power. I know this is very easy, guys. This is very easy. They have very highest catalytic power. Catalyze their substrate into their products. The next thing is they are highly specific towards their substrate. Guys, these properties are very important. Okay, don't take it easy. Don't take it easy. Next, most of the enzymes are protein. We know that. Means enzymes are made up with, I previously discussed about that they made up with tertiary and quaternary protein structure. I already described this thing. What is tertiary structure of protein and what is the quaternary structure of protein? Okay, inside our body, these enzymes work as a biocatalyst. Biocatalyst. Inside body, we call it as a biocatalyst. Human body, animal body, living things body. What they do, guys? What they do? They increase rate of reaction without itself being changed. Yes, guys, they don't change their self, but they increase rate of reaction. That this is the substrate and they want to convert into the product, and enzyme will add, and this what they do, enzyme what they do, speed the reaction. If this phenomena take 1 hour, then enzyme what they do, they speed this reaction, they complete this reaction in 10 minutes. Alright, guys? Alright or not? Guys, whatever I explaining here already written here. Alright, now next property is, I'm writing here, enzymes are protein with high molecular mass. Enzyme have high molecular weight because they have weight, guys, because they made up with protein, and protein are macromolecule. Simple. I already discussed this thing. What are the micromolecules? What are the macromolecules? Okay, next property is they catalyze biological. Yes, guys, there are inside our body, all phenomena occur with the help of the enzymes. Guys, inside the body, all kind of phenomena is done by enzymes. Okay, now they also affected here. They also affected by temperature. If you increase the temperature, they affected. If you increase and decrease pH level, they also affected. Now, guys, look at here some reaction, guys. These are the substrate and they change it into products. The substrate they convert into products. Okay, assume, guys, this reaction is hard for you, but you are familiar with this reaction that is occurred in our RBC. This is phenomena, this reaction going to occur in our RBC. What happened in with our RBC? That CO2 come from outside, from environment, and we intake. Of course, guys, some gases we intake, some gases we release. Okay, so CO2 present in the tissues, CO2 is present in RBC, combined with water, and they change into carbonic acid, and now they supply towards the body. Now they supply towards the tissues. So CO2 and water, they change into carbonic acid with the presence of carbonic anhydrase. This is the very fastest enzyme. This is the very fastest enzyme. Look at here. In the absence of any enzyme, this reaction is very slow. If I remove this enzyme, then this mechanism is very slow. With about 200 molecules of H2CO3 being formed in 1 hour, this reaction takes place in 1 hour. But if you add on this enzyme, this phenomena going to occur in every second. Now, guys, Another, you are very familiar with glycolysis. Glycolysis, this phenomena occur in our cytosol. Glucose change into pyruvic acid. I know guys, this is the 10-step process, I know. But each and every 10 step, they require enzymes. For example, like glucose, they want to change into glucose 6-phosphate. So hexokinase will require. Of course guys, each and every step, I told you now, inside our body, each and every step, requirement of enzyme is compulsory. This is the 10-step phenomena. Glycolysis is 10-step phenomena. Each and every step, they need enzyme invertase, isomerase. Without enzyme, this phenomena can't occur, not possible. And what is the special feature of enzyme? What they do? They change substrate into product. Simple guys. They change substrate into products. In our properties, I also told about this. Clear guys? Now, understand this graph. Understand this graph clearly guys. Be focused. Look at here. Here they made progress of reaction in this direction. Potential energy. How much energy required? This is the substrate level. If substrate want to change into the product, if substrate want to change into their product, what's going on? If without enzyme, we want substrate change into the product, then this parameter going on. They take highest transition state and then they convert into the product. No guys, this is not touching here. No, don't touch here. And now with the presence of enzyme, look at here. This is the transition state and yellow. Instant product will form. Means they lowering the activation energy. So what enzyme will do? Again add this property. Lowering the energy of activation. Look at here. Your point is forming. Clear guys? Here is some inorganic. Wait. Here is some inorganic term is also used here. Inorganic term is also used here. What is the meaning of inorganic ions? What is the meaning of inorganic chemical reaction? We know that what is enzyme chemical reaction. What is the inorganic chemical reaction? Enzyme we discussed. What is the inorganic? Guys, inorganic chemical reaction occur outside the body. Okay, these are not biological. They are non-biological. Okay, they are non-biological. And they are very simple, very low molecular weight. This is the you can cross-examine. Means this is the property of enzymes and enzyme reactions, and this is the property of inorganic chemical reactions. They are low molecular weight. They are micromolecules. Okay. Now, they are not specific for their specific in nature. They are not specific in nature, and it does not affected by temperature and pH. Means this is our opposite from enzymes. So this is your graph. Concept of activation energy guys. Here is some steps that enzyme will follow. First step is the substrate binds to the active site of enzyme, fitting into the active site. Means assume this is the structure of enzyme, and here another enzyme, and here another enzyme, and this is the structure of substrate. This is the enzyme, this is their active site, this is their active site, and here their active site, and this is the substrate. So of course you know this substrate bind with which kind of enzyme? This enzyme, I repeat, now you can identify this enzyme's nature is specific for this kind of substrate. So this substrate fit into the enzyme's active site. Now binding of the substrate induce the enzymes to alter its shape, fitting more tightly around the substrate. Now after fitting, wait, I draw for you. After fitting with substrate, the binding of substrate and enzyme more tightly. The active site of enzyme, now its close proximity of the substrate break the chemical bond of the substrate and the, okay, now what's going on here? Enzyme, what they do? They break there some chemical composition. They break there some chemical bonding. Now the enzyme release the product of reaction and free the enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. What they do? Now they break their chemical bonds, they break there some inorganic bonds, they break there some covalent bonds, and they change into product, but enzyme remains same. This is the product and this is enzyme remains same. There are some factors, guys, they affect enzymes activity. This is first temperature and here is concentration. This is the graph A, this is for pH, this is the graph B, this is for temperature, and this is graph C, concentration of substrate. If we increase pH, then at a specific time the enzymatic activity show their higher efficiency, but after unnecessary pH decrease the activity. After increasing slowly temperature, the enzymatic efficiency also increase, but at a peak they decrease in their nature. After increasing the substrate, means there are lots of substrate, and here is enzyme. With the starting of this reaction, every substrate bind with enzyme, 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 and when they are fully saturated with enzyme substrate complex. Okay, here this is the Vmax by 2, here Vmax and here Vmax by 2, here 50% of substrate will satisfy with enzymes. Okay, so temperature and pH and concentration of solute also affect what? Also affect the efficiency of enzymes. Some enzymes, I'm explaining in deeply, guys. Now listen to me. Be focused. I'm explaining this thing. This is the active site. Okay, here is binding site, binding area of substrate, binding area. This is the site of which? Catalytic area, catalytic area. This is the site of cofactor, binding area. Other than active site, there are many sites are there. We call it as a allosteric site. Other than active site, these are allosteric site. If some, I repeat, if some positive factor, effector attach, then reaction goes superb. Move further, rate of reaction will increase. If negative effector will bind, then the decrease rate of reaction. I hope this is clear. This is the binding area of substrate. I'm writing here substrate. This is the activation site, catalytic area. This is the site of where cofactor will bind. Clear, guys? Okay, let's take quick view of what is cofactor. Isn't your book or not? Okay, here we will discuss here. Okay, guys, after some time we will discuss. There are some classification of enzyme here. First is oxidoreductase or dehydrogenase. These kind of enzyme, what kind of property they have? They have reduction and oxidation phenomena. They do reduction and oxidation. The example is lactate dehydrogenase. Remember this enzyme's name. Now transferase. What they do? They move chemical group to one group to another group, like lysozyme. They transfer one group to another group. Now next is hydrolase. What they do? What is the meaning of hydrolysis, guys? What is the meaning of hydrolysis? Means there are some breakage of bond. I'm writing here bond cleavage with transfer of functional group of water. That's it. What is the lyase according to their name? Lyase, not ligase, lyase. They break. Oh, wait. They break bond. Okay, guys. These are hydrolytic bonds. They break, and they are non-hydrolytic, guys. Non-hydrolytic. All right, or not? Example is fumarase. Now next is isomerase. You can familiar with this example in the case of glycolysis. Remember, guys, when glucose 6-phosphate change into fructose 6-phosphate change, this group will change into this group. Only glucose change into fructose with the help of isomerase. What they do? They isomerize. Isomerization. Change the conformation. Okay, now next is ligase. Ligase means they bond something, they attach something, they join something. In DNA replication, DNA replication, transcription, translation. There are huge role of ligase in molecular biology. There are huge role of what they do. They synthesis new covalent bond. So these are the classes of enzymes, guys. First is oxidoreductase, dehydrogenase, transferase, hydrolase, lyase, isomerase, and ligases. Now let's move. Very important, guys. Question forming area. What is cofactor? Cofactor is very necessary for what? Very necessary for enzymatic activity. Very necessary for enzyme. Without cofactor, enzymatic activities not goes better. They need cofactor. And what is cofactor? They are non-protein molecule. They are non-protein molecule, guys. Cofactor will divide further into two parts. First is organic. We already discussed what kind of organics, and non-organic means inorganic. You know na, guys, you betterly perform how you perform to isolate organic molecule, and how we found inorganic, we get ash. Okay, remember, no guys, before starting this chapter we already studied organic and inorganic thing. Now don't confuse. Okay, inorganic means we also read down some ions, multiple ions. No, in organic they contains ions, and organic what they contain some carbon, hydrogen, and oxygen groups. They contain some proteinish part, they contain some lipid part, they contain some amino acid part, and they contain ions. Simple, guys, simple. Just I commonly that we use like salivary enzyme, salivary amylase. What they do? They change starch into simple sugar with the help of salivary enzyme. And salivary enzyme need what kind of inorganic molecule? They need zinc. Clear or not? Yes. Glucose change into glucose 6-phosphate. They also need magnesium. We know that they need hexokinase. Glucose change into glucose 6-phosphate. I know they use hexokinase. This is them, but they also require magnesium. And what are the organic molecules? Means they are organic. Means they may be protein, it may be lipid, it may be some amino acids. Clear or not? Organic cofactor further divided into two parts. Prosthetic group, already all things are written here. Prosthetic groups and coenzymes. Okay, organic cofactor divided into two parts. Prosthetic group and coenzyme. What is prosthetic group? What is prosthetic group, guys? What is prosthetic group? Simple. They are tightly bound to the apoenzyme. They tightly attach to organic cofactor. Example, look at here. Example is what? Like flavin. You know, you can hear heme, biotin. Guys, one of the most, biotin and avidin. These protein interaction is highest, strongest. They attach with the highest affinity. Okay, what is coenzyme? They are loosely bound with organic cofactor. The example is NAD+. Remember this example. Okay, guys, some vitamins in our body like vitamin A, vitamin B. So vitamin B have their multiple complex like vitamin B1, B2, B3, B12. So niacin is a part of vitamin. So niacin use what? NAD+. This is the coenzyme. Look at here, guys, already in your book. Okay, so that's all for now, guys. Your chapter is also complete. This is the summary. So subscribe my channel, guys, if you are not subscribed. Adhyan with Flow. And thank you for listening me. Thanks a lot.